to plasma cell differentiation, as well as autoantibody production. And the one thing I want to emphasize before moving forward is that this model, the TDNB crossed with the V6-LPR model, has fulminant lupus-like disease, but that the TDNB model alone, those mice are not sick. They simply have some autoantibody type properties, but are not actually sick mice that are a lupus model. Now, another model that I am going to utilize as part of this thesis is a VGA model, and this model has inexpression of MITF with no effect on any of the other MIT family members. And utilizing this model will allow us to clarify whether specifically MITF plays an important role in B cell to plasma cell differentiation, as well as autoantibody production. Maria showed Dr. Lopez-Ocasio showed initial data in this VGA mouse, showing that it does have splenomegaly and increased plasma cell numbers at 6 months. As a summary, the TDNB mouse has inhibition of MIT family members, all 4 of them, in specifically B cells, while the VGA model has inhibition of specifically MITF, but in all cell types. Now, to help clarify these different models and what we expect as we interpret the results that I will present for this defense presentation, is the fact that you can have abnormalities in specifically TDNB mice that you do not see in the VGA mice, as well as you can have abnormalities in VGA mice that you do not see in the TDNB mice. Also, there can be abnormalities that are seen in both. So the abnormalities that I will present in TDNB mice, the interpretation is that those represent functions of other MIT family members in B cells, but not necessarily MITF alone, because the VGA model does not have that same abnormality. When both VGA mice and TDNB mice have abnormalities, that supports a model where MITF is specifically playing a role in that function, because both VGA mice with only MITF mutation and TDNB mice with mutation with inhibition of all 4 family members have the same abnormalities. And lastly,